All right, so how did Mendel actually discover these three fundamental laws of genetics that all of genetics would be based off of today? So firstly, he started with an organism. He chose, as you can see on the screen, the pea plant. And it was good for a multitude of reasons. Firstly, they were readily available. He didn't have to travel for them. They were already growing where he lived. And two, they grew really easily. Not a lot of attention, not a lot of care required. And they grew fast. When it comes to genetics, fast turnaround of a species, of organisms within a species, is important. That's why genetic testing on people is not ideal, because we take so long to reproduce. Now, the sexual structures of the flower, because pea plants do have flowers, are completely enclosed within the petals, so that there's no accidental cross-pollination between plants. Right? We call reproduction in plants pollination, and when the reproductive cells from one plant ends up in another plant, then that's cross-pollination. That's what the petals of a flower are there to prevent. But it doesn't always work, and because it doesn't, we have a lot of genetic variation among plant species. Now, before we learn too much about Mendels experiments, I want to talk just a little bit about the basic structure of a plant, of a flowering plant. So flowers, all of them, contain both male and female reproductive structures. The female part of the flower is referred to as the pistil and produces eggs. It's located here in the picture. The male part of the flower is called the stamen, and it produces pollen. Pollen contains plant sperm cells, and these structures around the pistil are called the stamen. Now, when pollen is delivered to the pistil, the sperm travels to the egg, and as we know in humans and other organisms, fertilization occurs. The zygote is made. And here is the zygote. Seeds are plant zygotes. And again, fertilization produces a tiny embryo which is enclosed inside of a seed. So seeds are plant zygotes. Okay, now Mendel's use of pea plants in his genetic experiments. Now, pea flowers are normally self-pollinating. That means the stamen that we just saw in the last slide fertilizes the pistil found in the same flower. And again, genetically, in the species, this is important because it's going to maintain genetic stability, and also the pistil and the stamen, as you saw, are very close to each other, so it's easy to self-pollinate or self-fertilize. Now, the resulting embryos of self-pollination are going to have the same characteristics, right? Just like when an organism asexually reproduces, right? Although there's only one parent in asexual reproduction produces an offspring with all the same characteristics. However, in this case, this is sexual reproduction. Even though there's still only one parent, we still have sperm meeting egg, which is the definitive characteristic of sexual reproduction. But there, again, is still just one parent when it comes to self-pollination. Now, Mendel knew that these pea plants were what he referred to as true breeding, which means that if they were allowed to self-pollinate, self-fertilize, that they would produce identical offspring to themselves. And I'll give you an example here. If allowed to self-pollinate, tall plants would always make tall plants. Plants with yellow seeds would always make plants or offspring that produced yellow seeds. Okay, true breeding, always making identical offsprings to themselves. Now, these true breeding plants were the cornerstone of Mendel's experiments. Now, Mendel wanted to produce seeds by joining egg and sperm from two different plants. He wanted to see what would happen if plant A sperm cells were mixed with plant B sperm cell or egg cells. What would the resulting offspring look like? They couldn't be identical copies of an identical copy of the parents, because the parents are different to begin with. So to do this, he had to first prevent the possibility of what? Correct, self-pollination. So to stop the possibility of self-pollination, Mendel would actually remove the pistil, or the male parts, from one flower and remove the stamen, the female parts, from the other. Now, Mendel, like I said, cut away the stamens. Oh, I'm sorry, I had that backwards. Mendel cut away the stamens. Yes. Stamen is the male reproductive part and pistil the female reproductive part. I'm sorry if I said that backwards earlier. And then he would take the pollen from one flower and dust it onto the other flower. Mendel basically doing what bees do, right? Bees cause cross-pollination by picking up pollen from one flower and dropping it off at another. Mendel is playing the role of the bee in this case. And we would call this cross-pollination because it's a cross between two different plants and produces offspring from two different parents, just like when humans reproduce. Now, with this technique of removing the male structures, stamens, and the female structures, pistils, from opposing plants, Mendel could easily crossbreed plants and experiment with different characteristics. Now, before we move any further and look at the actual crossing of these traits and track the traits through generations, we have to know some terminology. Before we proceed, we're going to learn that P generation means parental generation, the parents. F1 generation refers to first generation of offspring, okay, so the parents' children. F2 generation refers to the second generation of offspring, namely the first generation's offspring, okay? And lastly, the term hybrid refers to offspring with parents of different traits. We are all hybrids. Your mother, your father have different traits, making us hybrids. Catch you in the next one.